In this video, we're going to do a weekly revision for weeks 1 and 2 of semester 2, animal reproduction. What is reproduction? By definition, reproduction is a process whereby all living organisms produce offspring. Reproduction is one of the essential functions of plants, animals, and single-celled organisms, as necessary for the preservation of the species as eating is for the preservation of the individual. So this is a general definition of reproduction. We have two types of reproduction in animals. We have the asexual reproduction and sexual reproduction. We're going now to see the definition of each type and later the differences. In sexual reproduction, we have male and female pairs. During sexual reproduction, we talk about fertilization, where one sperm coming from the male unites with one egg cell or ovum coming from the female to form a zygote. Then the zygote divides and differentiates into an embryo. In asexual reproduction, we don't need two partners, male and female. Only one individual reproduces on its own. Some unicellular living organisms, like the paramecium and the bacteria and the amoeba, reproduce in this way. Example of asexual reproduction in hydra called budding. Hydra is a multicellular living organism that lives in the seawater. In step number 1, the hydra develops a bud. It comes out of the main body of the hydra. It's called a bud. In the second step, the bud develops a mouth and tentacles. Tentacles are like false arms of the hydra. They're not real arms. In the third step, when it's fully developed, fully formed, it's talking about the bud, the daughter hydra detaches or separates itself from its parent. In the last step, the daughter hydra is now fully independent. A new hydra is formed. So this is called budding, and it's asexual reproduction because there was no fertilization and no male or female.
In hydra, a bud rises from the lower half. It grows in size. Mouth and tentacles are formed terminally. As soon as the bud becomes nutritionally independent, it constricts at the base and separates as a new individual.
Another example of asexual reproduction that takes place in bacteria is called binary fission. We also have 4 steps for this process. In step number 1, the cell elongates and DNA is replicated. DNA is the genetic information of the bacteria replicated, and it makes 2 identical copies of itself. In the second step, the cell wall and the plasma membrane begin to divide. In the third step, the cross wall forms completely around the divided DNA. In the last step, we have 2 bacterium cells are formed. So this is called asexual reproduction, binary fission. It takes place in bacteria, also in paramecium, which is also a unicellular living.
Some living things remain a single cell throughout their lives. For example, these are E. coli bacteria. Millions of these microscopic creatures inhabit our intestines. Bacteria are very simple creatures. Unlike most cells, a bacterium doesn't even have a nucleus. But when it comes to reproducing, bacteria are marvels of efficiency. When a bacterium reproduces, it pinches in the middle, and then the cell divides in 2. Each of the 2 new cells is exactly like the original one. Bacteria can reproduce very rapidly. Some types divide once every 20 minutes. At this rate, a single bacterium can become over 1 billion bacteria in less than 12 hours. This kind of reproduction is called asexual reproduction because only one parent is needed to reproduce.
I'm doing that.
Now we're going to talk about the sexual reproduction in viviparous animals, example, mammals. What are viviparous animals? In viviparous animal, the embryo develops inside the mother's body in an organ called the womb or the uterus. So the embryo is completely developing inside the mother's body. And the fertilization, which is the union of one sperm and one ovum, it takes place inside the mother's body in an organ called oviduct. So this is the viviparous animal, and we'll talk about one example, which is the mammals. Okay, like we said before, fertilization is the union of one sperm coming from the male and one ovum coming from the female to give a zygote. Steps of fertilization. First step, we have many sperm cells start approaching and surrounding a single ovum. In the second step, we have the one sperm cell penetrating the ovum. In the third step, the nucleus of the sperm unites with the nucleus of the ovum to give a zygote. And this is the process of fertilization. After fertilization, the produced egg or zygote will embed or implant itself in the uterus. Then the zygote or the egg starts dividing many times. First it divides into 2, the 2 divides into 4, 4 divides into 8, 8 into 16, and so on. It keeps on doing this until a complete embryo is formed. This figure illustrates the development of the embryo. You have the egg cell or the ovum. Once it's fertilized, it changes to a zygote. Then the zygote starts dividing into 2, 2 into 4, and so on until a complete embryo is formed. Now we're going to talk about the sexual reproduction in oviparous animals. What are oviparous animals? Oviparous animal, by definition, is an animal whose embryo develops completely inside an egg outside the mother's body. So the egg is laid to the outside, it doesn't stay inside the mother's body. Now we're going to describe the main constituents of the egg and the role of each one. We'll start from outside. The shell and shell membranes, they protect the embryo which is developing inside the egg. The yolk or the yellow of the egg is rich in lipids, and the egg white or the albumin is rich in proteins. Both the yolk and the albumin, they contain the necessary nutrients needed for the embryo to grow and develop. And we have the egg cell or the ovum, which later can change to a zygote if fertilized. And we have the air space, which constitutes a reserve of oxygen gas needed for the embryo to grow and develop. How an egg is formed. So first we have an egg cell and ovum. It goes down to the papillon of the oviduct, the top of the oviduct. Now the ovum will be fertilized by the sperm cell and changed to a zygote. The zygote will be surrounded first by the egg yolk. Then it will be surrounded by the egg white or the albumin. Then as it keeps moving downward along the long oviduct of the hen, it will be surrounded by the shell and the shell membranes, and then it will be laid to the outside. It must be noted that even if we don't have fertilization, the egg is laid. This means that the egg laying process will be done whether we have fertilization or we don't have fertilization. So sometimes we could have fertilized eggs which contain a zygote, and sometimes we could have unfertilized eggs which doesn't contain any zygote. So the process of egg formation can be done in two ways: if we have fertilization or if we don't have fertilization. When the embryo is developing inside the hen's egg, how does it take nutrients and oxygen gas? The nutrients are found in the yolk and the albumin, which we said before, they are rich in proteins and lipids or fats. These are the nutrients which will be used by the embryo during the whole process of development, which is for 21 days. And the oxygen is derived through the eggshell, which has small pores or openings which allow the oxygen to move from outside to inside. So this is how the embryo gets its nutrients and oxygen gas to grow and develop.